Hi, it's great to get a chance to talk to you. I'm going to tell you about the work that my team does in cancer data science. So, me. I got my PhD back at the University of Toronto in medical biophysics. And then a few years later, I went forth and got an MBA also at the University of Toronto. I'm a professor here in human genetics and urology, and I direct the cancer data science program at our cancer center and have a number of similar roles that are related to that. And, we're really interested in being able to take clinical samples, big data, information about treatment response, and pull them together into something that's going to shape the way we think about cancer and the way it's managed. And so to do that, that means publishing lots of papers. It means making sure that we have intellectual property to protect new drug targets or biomarkers that we develop, and collaborating really broadly in academia, where we're part of most of the major cancer consortia and have lots of one-to-one collaborations with specific universities, but also with lots of partners in tech and in pharma. And the idea there is to make sure that we're exchanging ideas, exchanging technologies, and coming up with ways to bring things to the maximum impact and the highest level of innovation. So what do I do? Well, effectively, I'm interested in cancer and big data. And the problem that most occupies me is the idea that patient outcomes are highly variable. This is a survival curve. At the beginning, every single patient is alive at time zero, and time zero is when they're first diagnosed with their tumor. Over time, many patients will die of their disease. And what I really want to solve is understanding that variable course, because if we do, then this group of patients, we can think about intensifying treatment, giving them more therapy. And this group of patients might be correctly treated or might even be overtreated. And we think about this problem in effectively every cancer type, every setting, every data modality. How do we optimally stratify patients into groups that need different therapies, and what should those therapies be? And so my research is all about turning data about this into insight, something that can inform practical decision making. And we kind of tackle it from three different angles. First is the question of early tumor evolution. What was going on before we knew the cancer was there? How did it get there? How could we have inhibited that or altered it? What are the features that direct that? Second, how do we make pragmatic clinical decision making better? Third, across that spectrum from pre-diagnostic to relapsed and metastatic disease, how do we integrate different types of data to create that insight? Different types of molecular data, here showing RNA and protein being integrated in novel ways, or different types of device data where we look at all sorts of mHealth. So I'll give quick vignettes on each of these. First, in the pre-diagnostic evolution, we established a multinational consortia to understand cancer evolution, effectively to be able to figure out what are the right methods to use and how should they be applied to this. We evaluated individual tools through a global crowdsourced challenge where we developed a way of identifying truth in cancer evolution questions, made a big data set available, and then had hundreds of people around the world work on it to identify optimal methods. And we were then able to apply those optimal methods to specific cancers to understand characteristics of how they relate to biomarkers. And so we would be looking at that to understand specific clinical questions. For example, we looked at a series of prostate cancers using that. We were able to stratify those cancers that were evolutionarily very diverse from those that were evolutionarily very simple. And it turns out at the top here in yellow, the evolutionarily simple tumors have very, very good outcomes after surgery alone. The red, evolutionarily diverse tumors have really terrible outcomes and need adjuvant therapy and need novel therapeutic development. And that gives you a lot of insight about how the evolution ties directly into biomarkers. But we also think about how to use really variable types of data. What are the algorithms that you can use to extract new information? And a great example of that is in a series of clinical trials of exercise, where we think about something relatively straightforward, where we look at patients who are undergoing a specific exercise protocol in the window between diagnosis and surgery. And here you're seeing real-time glucose, heart rate, step monitoring. We then link that to molecular features where we can take a look at individuals in purple immune cell infiltration and find out that exercise is changing the immune infiltration in the tumor. It's not only changing immune infiltration in the tumor, but it's influencing the immune composition of the blood. And we can link all of that together in network analysis to understand co-occurring sets of features from DNA to RNA, host to tumor, physiology to pathology and radiology, and bring those together to be able to make insights about what is going to work for an individual patient and what's the optimal way to consider management decisions. How do you take methods and figure out, do they really work? And we love auditing and optimizing existing methods and figuring out, are they reproducible? We develop and exploit advanced machine learning methods. We develop companion diagnostics. We understand how tumor evolution over time and space is linked to drug response. We figure out how to link clinical, mHealth, EMR, anything data into biomarker development. And we try to find things other people have missed by merging the advanced data sets that are generated here at UCLA on our patients with public data sets that everybody has access to. I'll stop here and give a moderately detailed vignette, and I'd love to talk to you about the work that we do, for example, in prostate cancer, but feel free to reach out to me or catch me at this meeting if you have any other questions.